came to Japan about 20 years ago. My background is actually on AI and robotics and things like that, and I've shifted into all sorts of different things. Now I'm actually working on bioinformatics, really, or system biology. That's the lab that we are basically in Japan. Okay, RIKEN, just to say something about my lab. RIKEN is probably one of the largest research institutes in Japan. So they have ample funding. The place that we are in actually is in Kobe, and one of the center which is more famous than we are is the RIKEN CDB Cell Development Biology Center. But we are actually, although we are attached to it, we're within it, but we actually belongs to another center called QBIC, which is a quantitative biology research center. So our work is mainly on developing technology, on like computational biology simulations, looking at whether we can simulate one cell kind of mathematically or simulate numerically on cell development, not cell development in itself, but on cell biology. But my lab that I involved with is mainly on C. elegans too. So mainly on developmental biology and on C. elegans, embryogenesis, etc. Now, going back into the development of our database. So I've just prepared a presentation as what Richard was asking. Try to see how do I get into the sharing. How do I share?
You might have to switch. Does he have to switch to the old version?
No. Yeah. Corner of my screen. I can switch to a closet. Oh, wait a minute. Oh, screen share. Okay, I've got it. Got it. Oh, here you are. Okay, I've prepared this presentation for you guys. So basically today's talk is mainly on using SSBD BDML formats to analyze and visualize embryogenesis on biological dynamics phenomena.
Ken, can you hold on? Can you hold on a second, because I don't see the slides yet. It hasn't transmitted.
Okay. The black doesn't work. Okay, wait a minute. Can you see anything?
Now I see them.
Okay, this is better. Okay, so I just use this one then.
Okay.
I don't know how Apple does it or whatever. Okay, so the motivation I've got. So basically, the reason what we want to do is on a quantitative, just to get numerical data from images. For example, like this one. Let's see what I can get going on. So there's an embryogenesis of C. elegans, and although direct observation is the norm, we're looking at it as well. Can the computer see it, basically? Nowadays, with image processing and computing powers, now we can actually extract different parts of an embryo. For example, in here we're actually extracting the nucleus of the embryo, and in 3 dimensions. And there are actually a lot of all this data around. This is zebrafish from Keller and fruit fly, which is on embryogenesis. And those are actually numerical data. Pardon?
The fruit fly, was that the imaginal disc or the whole embryo?
The fruit fly is the whole embryo.
Oh, it's whole embryo. Okay. Okay.
I'll show you later on more of it. but at the moment, what we found is that it's very difficult to use all this data. They comes in all sorts of different formats. It's difficult for us to locate them really. So the idea of this is actually to somehow to have a unified format for putting in all this data, and also to have a database that allowed us to do comparisons, to do more analysis of this data. Can you see the slide? I'll try to see what I can.
Yeah, it's quite readable.
Okay. So the project is actually funded by the support of the what we call the Japan's National Bioscience Database Center. It's part of a larger project on life science database integration in Japan. So they're basically trying to get all the databases in Japan on bioscience together into one entity, some sort of, such that we can people can search as well as can do more with it. So in some ways, it's supported by the government and with the ministry. Actually, basically. All the researchers in Japan have an obligation to support the project. So there's a lot of support in terms of getting data and for other people to use the format and etc. That's within Japan. So we are hoping that we can actually share this for the wider world. So our aim is basically, as the funding goes, the aim is to store and share all this different type of biological data to the community, and basically people can access it directly. Can also use different type of tools and data visualizations and analysis. Okay, any problems so far? Any question?
Does this? Okay, to what extent does this constrain people?
It doesn't actually constrain anybody. I mean, we actually still have to actively asking people for data, and they don't actually need to give it to us, but they're obliged, with their funding.
Oh no, that's not what I meant. I meant in terms, if one creates databases, sometimes some things just won't fit.
Oh yeah. So,
that's a political question.
Okay. The technical question is actually yes. When we design the, I'll show you later on. For example, like the BDML, what we've done. What happened now? We have people coming to us and saying that, hey, we don't have any, how to say, dynamical data. So its time point is equal to 0, so to speak, or time scale is equal to 0. So it's a bit odd sometimes that things happen, and sometimes, for example, like in our format, we actually have organisms. So we actually state some sort of organisms. But however, some of the simulations that some of our colleagues does, they don't have any. Is non-descriptive organism. Probably can be human, can be any mammal. So sometimes we can get into that type of. The idea is that we can extend those things, hopefully, and can adjust to it. But yes, we do find from time to time different requests that we haven't thought of, so to speak. Okay? Any more? Okay, so I'll carry on then. Okay, so just so what we've come up with, as what I've said earlier, is about this creating a basically a unified language in us, or what I would say is a file format that we can actually encode all this type of different data. So we come up with what we call the BDML, Biological Dynamics Markup Language. It's just another XML languages. It basically can represent 5 different types of objects, be it a point, a line, a face, a circle, or a sphere. And you can then define it within it, as in here, in the slide. With that, we were able to actually represent, have a flexibility of represent different type of things. from molecules, cell divisions, to C. elegans behavior, to nuclear dynamics, and et cetera, in different scale, and for different organisms, for simulations and measurements, etc. So basically, we put them all into a database called SSBD, and it is open access to everybody. So if you click on this website, you can get to it. And I will actually do a demonstration on some of the parts of it. So you can actually get some of the data sets of both quantitative data, for example, numerical data that we actually extracted, for example, from image processing, and also by hand. Quite a lot of those things are actually, although I've done extracted automatically, after that we have to actually check by hand, eliminate some of the errors, and etc. etc. And we also have image data. Some of the data actually comes with the image data, so you can actually look into it. And we actually provide some of the tools for 4D visualizations as well as analysis. Okay, don't know any of you are interested in this. Just quickly go through. Basically, is internally is just a SQL database with application framework. I don't know. Are you aware of the Omero database system, which you can store images? We actually utilize this. This is also open source software developed by the University of Dundee in the UK. And so far, I think this is more interesting. What is currently available on SSBD? So we have E. coli, single molecules, but we also have a lot of C. elegans, nuclear experiment from Bio, and Kyoda, which is one of my colleagues. He's got 180 sets of RNAi C. elegans embryogenesis data up to, they're using, well, I'll show you later, it's actually using DIC, so it's only up to about 32 cell divisions. And also we have others from Toyota and things like that. And we also have simulations. Kimura actually do some simulations on C. elegans, pronucleus, microtubule, and things like that.
And what is this entry number ML? What is it?
Entries number means that it's the number of entities actually available within it. So, for example, this I'll show you here is that, for example, there's the number of nucleus and things like that is been extracted and stored as an entry.
Column to the left of that, what is that?
This one?
The BDML.
BDML is the file, so you can actually download the file set with the file that I mentioned. So one file, for example, one file in here, 186. So there's 186 sets of data.
Oh,
so the one file is a data set.
Okay, gotcha. Okay.
Fruit fly. And the zebrafish is also very large. So they actually have a lot of nucleus, and it track all of them. And so each of them are an entry in the SQL database, if you think about it that way.
Which Keller is that?
Pardon?
You know the first name for Keller? Hello? Ken, can you hear me?
Sorry, I lost you.
Oh, you did? Okay. Yeah, I figured. Do you know Keller's first name? I'm trying to figure out which
Keller.
Keller.
Yeah, Keller. I need to find for you. Sorry. Wait a minute. Keller. Wait. I haven't met him. Wait a minute. P.J. Keller. So
okay. So
Philip Keller. Sorry.
Philip Keller. Okay.
I'll send. Well, I can drop you some of his references.
Okay.
So he written in Science and some others. Genesis.
Okay. Now a stupid question.
Yeah.
How many of these data sets identify the cell as the cell type?
Actually, quite a lot of them. For example, Kyoda, the data set itself have both the annotation of the cell as well as the lineage. So you can actually track. The data comes with tracking of lineages.
Tracking of lineages. Okay. And
so I'll at the moment I will show you further. So what we have here, we can actually I'll show you the website, and I think it may be easier to for you to see how it works. Where is it? Okay, I need to do a share different. How do I go back to it? Stop screen sharing, and then screen share again. Okay. Now I lost my own my own page. Yeah. Okay. Here is the. Can you see the website? This is the SSBD cubic website. This is a C elegans by one of my colleagues, Kyoda. So what he did, if you click on it, this is actually a wild type, and if you click on the view, you can actually see the visualization of it, and this is actually. Sorry, I clicked it too far. I clicked too far. Anyway, so you can actually scroll it and to see how it's split and how the nucleus is extracted together. And you can also see it's in 4D. So you can actually see, you can actually zoom in and see the actual data set, how it was connected. Now, I go back, sorry about that, I go back and before the summary page, you can actually see the actual images. And you can actually...
Hold on a second.
Somebody is calling.
Oh, that's me. Just a second.
Oh, hi Dick, how are you doing?
George, yeah, I'm on another call now.
Oh, great.
Okay. Yeah, we had to cancel the other one that I told you about. Okay?
Okay.
Okay, so let me get back to this other phone call, okay?
Okay, no problem.
Okay, thanks, bye.
Bye-bye.
Okay, sorry guys.
That was I had an incoming call on Skype.
It's all right. I was just trying to say, you can actually, sorry about the screen size, you can actually see the actual EIC image data set. So I'm loading all the data sets. These are all available for you to download, or you can view it on screen or whatever. And as you can see how each slice of the images are extracted. So if I click on this one, I don't know whether can you see this one? Not exactly see what you will be able to see, but actually you can actually see see the actual microscopy image, and with that we extract each of them. We extract like as a line, as an outline of the nucleus into a line, and you can actually see different layers how it's been extracted. And all this data are actually becomes a numerical data in the database, which you can access and been saved. So all this data, unfortunately, this visualization have not include the annotation data. So the annotation data is actually available on the data set. And on top of it, it also have lineage you can play around with, which we haven't actually get into displaying that. But one of the tool sets, if we go into another tool sets, the standalone tool sets with the BDML viewer actually provide you with some of the lineage data available. Okay. Anybody have any questions?
Okay, the serial sections that we see are light sheet microscopy or what?
No, it is a DIC.
It's DIC.
Yeah.
Oh, okay, in the living embryo. Okay. Okay.
Yeah.
DIC. Okay.
Yeah.
And, what's the? Do you know what a typical separation of the sections is?
Wait a minute, 0.5 micros. I can't remember directly. We have a slide of 66 slides.
It says half a micron. Okay, I'm reading it.
Sorry.
There it is. Okay. Okay, it's half a micron. Okay. Okay, which is about the limit of light resolution. Okay, so that's reasonable. Is the DIC data corrected for blurring? You know, when you do light microscopy, your vertical blur is greater than the horizontal. Is this deconvolved for that?
Yeah.
It is.
The vertical blur. I didn't actually do the actual experiment, so I need to refer back to my colleagues for you, and I'll get back to you later on.
Okay.
So the vertical blur is greater than the horizontal one, you said?
Yeah.
Okay, I'll check that for you. I can't.
Typically by a factor around 2 or so.
Okay.
But there are de-blurring algorithms for correcting it. I'm just wondering if this has been de-blurred.
Okay. The vertical blur.
Okay, now also these are contours that we're seeing, superimposed contours from multiple sections.
Yes, it's basically we extract each segment, and then we bunch them together into a database as 3D. So you can see now it's actually the layers, contours of a nucleus.
Okay, can you have the ability to show just one section?
Well, yes, you can access it. At the moment, the visualization tool does not.
It's not.
I mean,
okay.
Of course you can. I mean, it's just data, and I'll show you later on with the Python data tools that you can actually just extract whatever parts.
Okay. Okay.
So I mean, it's just data. It's not a movie, so you can actually.
Looks like it's not smooth, like it's raw.
No, it's not because it's got all sorts of different things in it. Can you see Can you see this page?
Yeah.
Yeah.
Okay.
Yeah.
So and can you see the embryo when I click on it now? I've got to see the whole embryo. No.
No.
Okay, I'll switch. So wait. Stop sharing and share this.
Okay.
Yeah.
So you can actually, this is from Omron, which comes with the system. Wait a minute. Oh, God. No. So you can actually look at different slides on it, I believe. And so that's what you say about blur. It you may be able to see that in the actual image.
These are DIC. I can see these are DIC images. Then.
Yeah, that's the DIC image. Yeah. So what we are doing is just we are kind of extracting the outline of the nucleus, and with that, sometimes it's not actually smooth. As what I mean. Sometimes you just and we just leave it as that, as an outline, rather than saying that, have a definition of what the outline, whether it's a smooth one or not a smooth one, or what.
Right. Because whether a protrusion is real or an artifact of the
so
obvious.
Yeah. So all this data is available. So you can actually click on it if you're in your computer. You can click on it and play around with it. It's not. You can download it or play around with it. Okay. Let me get back to the other part. Where are we? Go back to my thing. So actually, so what we have now is that we can actually we can actually show different quantitative data. Just now, I just show you the C. elegans in DIC. You have Bao, which is a GFT extracted data set of C. elegans, and this is more. Maybe I should show you this one too, because I think you guys are interested in it. Go back to share. Stop this. Share. So if I go back in here, and if I say search for Bao for C. elegans, we've got some data sets. And this data set is 195 time point, so it's much larger. And as you can see, it actually gives you more, because the Kyoda one, because of DIC, it can have a limit on of all the nuclear or the cell division that it can actually record. So this one actually gives you a more complete set of embryogenesis of C. elegans.
And these are all smoothed out.
This is actually, because it's actually a GFP, so they actually extract the center point of a sphere,
okay.
And they actually give a radius too.
Okay.
So so there's no smoothing or not smoothing. It's just that it actually is a GFP, so it's just a blob. Okay. And with that,
but the data is the data the same? It's these DIC images is the raw data.
They are different. The format is the same. They're different type of data.
But here, what they're doing is replacing the nucleus by a centroid and an equivalent sphere.
Yeah, exactly.
Okay.
So although they are in the different, in the same file format, the data can be all different. And yes, those are different data. They extract it differently. They do different things on it. The DIC, because it's live, it's not. It doesn't have a GFP, so you can say that the wild type is wild type, whereas this one.
Okay.
So you got different type of data. You can look at different things.
Bradley, is this the data you use to get the volumes? It is. Okay.
Yeah.
Okay.
So it's same data set from Bio.
See, what we were doing is classifying each nuclear division. Or each
Okay.
well, these are nuclei. These are nuclei, not the cells.
Yes, that's right. So at the moment, for example, the DIC one is also nuclear, the data set. But however, because we provide with the images, hopefully somebody else will be able to reuse it and to kind of extract the, say, membrane to get a cell or whatever, all of it.
Can you show us some raw data to see whether or not we can see the cell membranes?
Yeah, I mean, that's another, I mean, some of my colleagues are working on those things. So it's to see whether we can extract cell membranes.
Okay.
At the moment, it's quite difficult from DIC. So we are trying to use GFP and other things to extract that.
Oh, you're trying to get a label for membranes?
Yes. But
Okay.
at the moment, we don't have data set for that. And we're still working on those.
Okay. So would this be a membrane protein that they're trying to tag?
Yes, that's right.
Okay. Okay, that would be excellent because what we really need for our analysis is the volumes of the individual cells.
Exactly.
Those are hard to approximate from the nuclei.
Exactly. So hopefully, I mean, so the whole concept of it is that the database is available as a resource for further investigations, and hopefully somebody can utilize it better than us, can make use of it and find new things, etc. Okay, good. I'll go further then and carry on with my talk. Time-wise, are you guys all right?
Yeah, fine.
Okay, good.
We have all evening. We have all day.
Now, how do I do a share? Okay. So, accessing the database. So I'm just show you how you can access it on a kind of website basis. The other one is that you can actually retrieving the BDML files. You can actually download the files. I'll just quickly show you where you can do that. So if I go back one page before you look at the viewer, you God, die on me, does it? Doesn't actually do anything. Well, sorry, something wrong with the connection. Hello? You're still hearing, guys? Okay. My computer is playing up. Okay, sorry. Okay, so if you go in here, when you see the images, you can actually have a download image, download BDML. So this is the actual data file. Download images, you can download the image as zip files. View it, you just seen it. And you can actually view the images here separately. So just quickly about, you can download the images. Now, the other way of doing it is, I don't know whether you guys are familiar with it, is to actually programming through a REST API. So, sorry. Oh, God. Stop this. Share this. So you can actually go through a RESTful API, and you can actually, for example, in using an API, you can actually use different computer languages or R or whatever to actually get the data directly from SSBD. So you don't actually need to download all of the data sets in order to view it. You can actually view a part of it. The API is given in our website here. You can have a look at it. I also actually put in a Python example of using that on GitHub. So you can actually have a look at GitHub in OpenSSBD, PySSBD API, or if you use Java, you can do that. For R, MATLAB, or other things, is similar. Now I'm just going quickly to show you how you can actually extract those things from Python. See what do that.
So your viewer incorporates the REST API?
The viewer actually uses some of the API. It uses, but it doesn't use all of them. I'm planning to replace all the stuff with API, so you can actually use that too.
Okay.
At the moment, it doesn't. It actually uses more than that to the browser viewer. But I'm actually showing you is that you can actually do a similar view, not as interactive, with just the API. Okay, so sorry. This is the Python API to get into the database. I'll just restart and I just continue running it. So I just load all these modules, reloading it. And one of the things that to display 3D is use a called iVisual, which is a bit unusual, but it's in there, so you can try it. And then you can load JSON, and then you can load the API itself, and then after that you just define it. And now by using this one, I'm just loading a simulation, and then it can actually display the result as an API as the data. So I'm just showing you the result data. So you can show you all this data. This is actually grabbed from directly, interactively from the database. Of course, you can search for different things. I'm just showing you about whether I can API search for data. So you can actually see all the data. So I need to load this function. This is actually to load the function to get all the coordinates at a specific time point. So here I choose a time point. So I'm loading it. So you can actually see that I should load time point 15, and those are data set X, Y, Z, and T. And I think this is the bio data set, the one that you guys also have download. And by doing that, I can do I need to load the scale to look the scaling on T and Y, X, Y, etc. And I just set up my canvas, and then I am going to plug this up. So here you are, time point 15 of C. elegans. See what I can control, and that's in 3D. So you can actually zoom it, and those are data from the data set. Okay, so just one time point, but it's very simple and easy that you can just do that, right. And it's all in GitHub, so you can actually take a look at it.
And suppose we wanted on that display to connect those nuclei that are from daughter cells from the same mother cell. Draw a line.
Yeah, you can draw a line onto it. I think it actually comes with. I'm not so sure about the Bao one. The Kyoda one come with a link called. It's called I've searched it through for you. It actually comes with annotation as well as the linkage, the lineage. Is it lineage? The tracking data, basically.
Okay. Okay.
So some of the data set come with the tracking data, some of them doesn't. Okay. Depends on the data provider. But yeah, and you can actually do a prolification graph. So you can actually count the nuclear, count the number of cells stage through a time point, etc. So you can do all sorts of different things with it. Okay, so I'm going back to some of the things that we are doing. So not only that, we also actually in our website you can actually download a ImageJ plugin tools that you can actually, if you have images, you can actually do your own BDML. At the moment, it's still the saving the BDML is still, I think, is still under to be released. But some part of it is already there, and you can actually see visualization. There's a standard kind of plug-in ImageJ plug-in visualization to look at the BDML file. So if you download the file or if you create a file, you can actually look at it independently from a browser or from or from the database. I've mentioned about some of the analytical tools. This is written in C, Finochar. So you can just look at some of the position, volumes, distance, displacement, some of the functions there to give you those type of things. And with that, you can then actually create different analysis. This is one of the things that we did just to kind of showcase how those things can be used. For example, in C elegans, you can see the kind of synchronous divisions, whereas in zebrafish, early on it has that property, but later on it kind of changed. Fruit fly seems to be a bit different. So it's quite a lot of things that you can actually, from the data sets, you can actually see a lot of things that you normally would not be able to find. To see.
The fruit fly probably showing some apoptosis.
Yeah. So, so some of the things just you can play around with. Okay. What happened with SSBD is that we also open source the software for saving all these things. So we actually put it into a GitHub called Open SSBD. The only thing difference is that it doesn't include the OMRO. You need to kind of install it separately and to link it separately. The difference is basically it runs the SSBD currently is run on Red Hat server, whereas OpenSSBD run on Ubuntu. It has some limitations reading different version of BDML files. At the moment, it doesn't support phase yet, but hopefully we'll put it on soon. It also does not support multi-part BDML yet. Browser visualization, the visualization that I've shown you on the browser at the moment is only for SSBD because I actually need to tweak it manually to sometimes it's actually I have to lower the resolutions in order to get a smooth data sets to on the screen. Whereas for this one, so the browser visualization is only on single time point, so it's just to make it easier. I also have a new read file API to read in files. So you can actually also play around with it if you have data, you want to create your own database, and you can use Docker to it and things like that. So how does SSBD and OpenSSBD can be used together? What we are seeing is one of data bazaar and one of data factories. So we are kind of SSBD. We are going to accumulate and store all kind of big, large data sets, genome-wide or genome-scale data sets. Whereas OpenSSBD can be more kind of research groups. Somebody wants to share some data, they can do their own. Now the problem at the moment, we don't have a solution yet. But how we'll get into the same problem that I mentioned before. How do we actually be able to search all the data across the internet, so to speak? One of the solutions that we are working on partly with our funding agency is using RDF. So it's using RDF formatted data. At the moment, the RDF data of SSBD is actually available on other database. Not so ours is just only on the endpoint. So on the RIKEN database as well as the another database here. So you can actually look into more things by looking at the elegance. You can maybe able to find more interesting things, the genes, etc., etc. So we hopefully using RDF or similar technology, we can link different data sets together. This is just some of the one of the ways of using RDF and Sparkle in order to search across different databases. So there's 3 databases to search across, and you can do it by using like SQL-like query language. Okay. Any questions so far?
Yeah. How much in terabytes? How much data is stored now?
We have something like a 300 terabyte server. At the moment, it's about less than 100 terabytes, about 50 terabytes thereabout. But we're expanding mostly because of images. So we're getting more and more large images. Quantitative data is still difficult to get people to do quantitative data as well as to give us the quantitative data. We hopefully can add more. There's 2 sets which I mentioned earlier, which may of interest to all of you, which hopefully will be released either within this year or next year. Which is 2 of my colleagues, they are doing it. Kyoda is actually have a genome-wide knockout RNAi data set, about 1,000 data sets by 360 by 666. So 66 slides, 100 and 360 time points. I think that's wait. 360 no 1,147 data sets. 360 time point each, I suppose. And Tohsato is quite interesting. She actually extract data from Phenobank, the movies of Phenobanks on genomic data, and then now it's become a quantitative data, and she did some analysis on it, and so that will also be released soon in the field, well, within a year or so. So those are two interesting ones. Other thing I forgot to mention, which is quite important, at the moment we have a policy for storing data. So all our data at the moment have to be published in peer-reviewed journals. However, we're starting to receive requests for data to be stored, which is, for example, like to be published, like just being reviewed. So at the moment, the policy is we only share, or we only store and share data that is published in peer-reviewed. It's just to ensure that we have the quality of the data. Just we say that, well, you can, if you have trouble with the data, you can actually go back to the journals or to clarify with the author. But maybe we'll change this in the future because of the request of to store, to have pre-published data sets. But at the moment, we're still basically all the data that are available have been pre-reviewed in journals. Okay. So the next part of what we are doing at the moment is we're actually expanding it a bit further by the request of our funding agency, that we are actually also going to store large microscopy images that have not been processed. So we may not actually have the quantitative data, but just the images. And mostly we are likely to be still concentrate on images that are dynamical. So, for example, embryogenesis or cell divisions or something, instead of just a single picture. Although we are not excluding them. So if you have kind of super resolution data sets and would like to store it, we'll probably store it at the moment. So we are going to expand the database into storing more images. Future plan of it, we are looking into changing some part of the internal system from an SQL database to a HDF5 probably. And we are also likely to more of a distributed architecture for the SSBD. The other part of it is that although BDML include RDF and ontology, SSBD, the database actually itself have not actually include ontology. That's part of what Richard was saying earlier about how do you adapt. I think, well, the point is we need to then have to add in new things, I suppose, in order to support those features. And obviously to probably do a little bit more and to on software tools on visualization and analysis. So in summary, the SSBD is a database developed and funded by by Japan, mainly supporting Japanese scientists in storing and reusing those data sets. But also we hopefully be able to facilitate and contribute to others to store and share data, and we can do more things with it. So here you are. That's the end of my talk. Hopefully that's not too long.
So the SQL database doesn't store the images itself, though, right? It stores the metadata.
No, SSBD database stored both the images, the quantitative data, and the metadata.
Oh, okay.
They store everything, but the images are stored in a separate system. Architecturally, it's stored in an Omron server,
Okay.
but it's integrated with it. Okay. At the moment, the API does not cover that yet. We are looking into it. And also we are looking into overlaying, for example, images in the visualization on the browser. We are thinking about overlaying the images on top of the quantitative data, or to have them side by side, such that you can have a more smoother to understand how the images related to the quantitative data.
Now, if you're transmitting the data uncompressed currently, or is it compressed? If you rest, for the REST interface, for example, does it decompress?
The data is actually in JSON uncompressed.
Okay.
So it's quite large.
Yeah.
And so in terms of visualization, I actually have to skim it down. So it's just only approximation. It's not a real one. Partly because of the browser problem and all sorts of other things.
A number of groups are working on segmentation of cell images from the cell membranes.
Okay.
incorporated in this in any way yet?
I haven't. It hasn't. There's another thing. It's quite interesting. We have discussed it, but we haven't actually. Well, the idea. Well, okay. A lot of groups are asking us about. Oh, can I store the images and you automatically image process and extract those things for us? And we say no. And so basically, at the moment, we do not actually have the capacity to do the image processing algorithm as well as the to be available as a software as well as a service. But maybe in the future, I don't know. But we haven't actually, in terms of how they incorporate that, we haven't actually have done anything on it. One suggestion is actually to have, for example, each data set will have some sort of metadata to tell you about how the data is being extracted, although it's already done in a database, on the publication in the paper. But we thought that maybe we can actually do a bit more to have a way in such that, as what you mentioned about Google AI and things like that, whether we can actually use deep learning or something like that to do more on data extraction and things like that. But at the moment, yeah, I think it's really a difficult area, quite challenging. But we haven't included in as a service in here.
Well, let me describe a data set that we're working on right now. Bradley's doing the most of the work on it. We have a Tanya Wolff published a poster in 1993, which was at the back of a book on Drosophila.
Okay.
And the poster was a hand-drawn image, probably using camera obscura. Every single cell in a Drosophila imaginal disk.
Okay.
Okay. Bradley's counted them, and they come to over 9,000 cells. Is that right, Bradley? Okay. Now, about 9,000. Okay. Now, this is a very remarkable image. It took a lot of work for her to produce it because it's drawn by hand.
Yeah. John Sulston, right?
What?
It's the drawing of John Sulston, is it?
No. It's by Tanya Wolff.
Tanya Wolff. Okay.
Wolff. It was a poster in the back of a 1993 book on Drosophila. Okay, I can send you the reference.
All right, thanks.
Okay. Okay. Now, what happened with this is my mother, who's an artist, about 10 years ago, went over this image in detail, and she closed all of the boundaries that were open.
Okay.
Okay, that had gaps in them.
Okay.
And she also removed all of the. Occasionally, we digitized the whole thing, so she filled in the gaps and she removed any extraneous points.
Yep.
Binary image. Okay, so the binary image shows all the cell membranes or all the cell boundaries of all of the. Okay.
Yeah.
And what we're doing at the time, back in the late 90s, with a couple of people, with my wife and Hillary Alto, we did a variogram analysis along the. You're familiar with the morphogenetic furrow in the imaginal disk?
Okay.
You know? Where's the?
Sorry, I didn't quite hear you.
Okay, let me get the chat going here. Chat. Where's the chat window on this thing? Where are? Chat. Oh, there it is. Okay. Yeah. Okay.
Yeah.
Okay, it's in the chat.
Okay, furrow of the eye. Oh, okay, interesting.
Are you familiar with this structure?
No, I don't. And actually,
Oh.
quite a my colleagues, research groups are actually doing in CDB is actually working on Drosophila. So okay.
Oh, okay, okay. Well, we would hope what we're doing inspires more observation because we have just this one picture. Okay.
All right.
But what the morphogenetic furrow is, it's a set of where the cells are contracting at the apical ends.
All right.
And, so that creates a furrow in the surface, and then the furrow moves slowly across the tissue.
All right.
As it moves, behind it, the ommatidia start forming. The little the groups of cells that form the tiny lenses of the fly eye.
Okay.
Okay? So I did a very crude analysis with my wife, Natalie Gordon, and with Hilary Alto, where we put one line down the morphogenetic furrow and then measured the distance, the intersections of that line with the cells. Okay? And then we did what's called a variogram analysis, and we showed that along the morphogenetic furrow, you had 2 large cells, 2 small cells, 2 large, 2 small, along the length of it. So this detected what we think is the very first pattern that forms as a result of this morphogenetic furrow moving across the tissue.
Okay, interesting.
Now, Bradley is doing further analyses where we're getting the number of cells, we're looking at patterns of the cells and various kinds of distributions, and we're analyzing these 9,000 cells to death in every way we can think of. Okay? And what you see is a progression of the evolution, the progression of the formation of the groups of cells that later form the lenses.
Okay.
Okay, now, is this kind of data set something that would go into your...
Yeah, we will be interested in data sets that, any data sets, we'll be happy to be included. But what we are more in, what we that's what we said. The pulse at the moment is if you have a peer-reviewed journal that that describe that, we'll be happy to.
Oh, okay.
To work with you, change it. We actually at the moment we actually do the conversion of your data sets into BDML ourselves. Obviously, we only have a few of us, so we can only do a few a year. So yeah, but if you have
the status of this is the variogram analysis is published in a book I wrote in 1999.
Okay.
And when we have something interesting, we'll submit a paper. Okay.
Yeah, great. I mean, even a book. I think if a book is a publication from our point of view, I suppose I need to check. I need to check. But if you have data, we'll be.
Well, and the original data was published. I mean, it was published in, as I said, it was a poster in the back of this book. So the original data was published.
Okay.
Okay. Well, then, Bradley, what do you think of working with Ken to make that a formal part of the data?
Yeah. Okay.
We can send you an image.
Yeah.
Show you what we got. And then the analysis, we're trying to analyze it and see what interesting things come out of it. Now, our hope is that once it's done, that this is just, of course, one image, one time point.
Oh, okay.
Basically, okay? It's like having one snapshot of the development of this tissue.
Okay.
But we hope that by doing this analysis, we can inspire people to start getting some time-lapse data.
Oh, okay.
Okay?
Okay.
And if you've got Drosophila colleagues, maybe they'd be the right people to do it.
Yeah, probably. If some of them are interested. Definitely of something of interest to us. Especially in morphogenesis and things like that. If you have image simulations. We don't need to be experiment. It can be simulation data.
But this is real data. This is experimental data.
Okay. So,
okay, let me describe another data set which we're working on getting.
okay.
I'm working with a former student of mine. Her name is Susan Crawford Young. Okay, I put it. It's in the chat there. What we're doing is developing a technique. Are you familiar with amphibian embryos?
Pardon?
Are you familiar with amphibian embryos? Frogs, salamanders?
Amphibian? No. But
okay.
yes, carry on. I'm not familiar with them. But.
Okay, these embryos are have a lot of yolk in them, and they're bottom heavy.
Okay.
Okay? So if you take the embryo and you turn it, it turns itself back right side up.
Okay, interesting.
Okay? Well, we're taking advantage of that by developing a robot to turn it upside down and then take it while it turns itself right side up again.
Okay.
Okay? And then this will give us a set of images of the surface, and then the robot rotates the embryo and flips it another way, and then it rotates another way. So the idea is to get images of the whole surface.
All right, okay.
Doing this, okay? And this would be on a live embryo, and we would do it in time lapse.
Yeah.
And then get the whole surface of the embryo over time from the single cell stage through at least neural tube closure.
Okay, that'd be interesting. Yeah.
Okay, so that's what we're trying to do. So this is the robot has just been completed. The software to run the robot has just been completed and works.
Okay.
Now we're waiting for the salamanders to lay eggs. Okay. We need eggs. Okay.
Okay.
Okay. We use an animal called the axolotl. Its Latin name is Ambystoma mexicanum.
Oh, right. Oh.
Axolotl. Okay.
Okay.
Okay, so this is another data set which might go into your thing eventually.
Yeah, that'd be quite interesting, yes. Definitely.
Now, an advantage of these is rather peculiar. The embryo we're dealing with has, I don't know if you know this term, variegated pigmentation. Okay? What this means is that the amount of pigment varies has very large difference from one cell to the next.
Okay.
So you can easily see the cell-cell boundaries because of the contrast between neighboring cells.
Right. So we can actually do some segmentation out of it, I suppose. Okay.
Probably can do segmentation. You know, we have to see. We've got a two-step thing. First of all, we have to get the robot to work, the embryos to cooperate, and get all the pictures. Then they have to be montaged into a four-dimensional data set.
Okay.
Okay.
Yeah.
So this is the surface of the embryo versus time. Then you do the segmentation, and then we do cell tracking, and also what we've observed on these embryos are waves of contraction and expansion of these cells. We want to be able to track these waves over the surface and analyze them, and how they're related to the morphogenetic movements, etc.
Okay.
And my wife and I have written a book which will be coming out in a month or two. Or I'll type it in here. Okay. And this book gives our theory of what's going on.
Okay. I'll order it.
Okay.
Is available or not available yet?
No, we are waiting to do the last proofreading.
Okay. Okay.
Okay.
Yeah.
And so it will be printed probably in July or August.
Okay. Interesting.
Okay.
Yep.
So this gives a theory of it. The basic idea is that we think that waves of contraction or expansion of the cells are the triggers for cell differentiation.
Okay. Yep.
Okay. And that's the fundamental idea. And
And you talk about wave. What wave? Calcium wave?
No, well, they would be calcium waves. They'd have a calcium component.
Okay.
And that's actually Lionel. Do you know the work of Lionel Jaffe?
No.
No. Okay. Lionel Jaffe. He died a couple of years ago, studying calcium waves.
Yep.
And he's published a little bit about the waves we observed, and he's predicted that they are calcium waves.
Okay.
They're very slow. He calls them ultra slow.
At the moment, what you're trying to say is that the morphogenesis, the wave that you observe as a pattern, is actually the cell division pattern. Am I correct?
No.
So,
no, it's the cell differentiation pattern.
A differentiation pattern, okay.
Differentiation. The wave triggers differentiation.
No. What is the wave?
The wave is a contraction or an expansion of the surface of the cell.
Okay.
It's an epithelial cell.
Okay, the cell, so you talk about the cell behavior causes a wave that do a differentiation. Okay.
Yeah, you got a propagation of contraction or propagation of expansion from one cell to the next. Okay. Now these waves they're very easy to observe, but we've never observed them in full 3 dimensions. This would give us an opportunity to do that.
Okay, that's interesting.
Okay.
Yeah.
It also, with the data, we can then start trying to analyze the mechanics and stuff like that.
Yep.
Okay.
Yeah. Yeah. I'm just reading. We were suspecting it, but I think the other group in Hong Kong have beat us to it. Is the time length, or actually Bao actually mentioned it too. The time length of the division of C. elegans actually kind of correspond to the differentiations.
Oh, really? Bradley, you got that?
the time that what's it called?
The duration.
Cell cycle. The cycle of the cell.
Cell cycle.
The length of the cell divide correspond to the cell differentiation. I'll send you the connection, the link. Wait a minute.
Okay.
Yep. Yes, I think it is quite difficult at the moment, and that's what we were talking about. This one idea is cell division timing. So whether we know about cell division timing of C. elegans, and we know about C. elegans differentiation is already kind of set. So the idea about is that whether we can actually, from time division, we can actually analyze, for example, other species in terms of when do they actually decide what they got the fate. When does the fate decide? And can we tell from different time divisions? That's one part of it. The other one is, personally, I'm more interested in the physiological side of it. Is when the cell divide, how much force does it require, and how does it change, and why does it spin around, and things like that. And with different species, you can actually see different things coming out. So yeah, I mean, it is not easy at the moment. It's not obvious either. How we can use the data in different species to analyze certain things. But I think because of the data is now available together in one place, it actually gives you a possibility of actually doing some analysis on it, whereas before it's quite difficult. So it can be quite interesting, I think. But I don't have a concrete kind of example that I can give you that what we can achieve out of this. Okay. Yeah. Okay. Yeah, I was wondering about the DevoWorm. I kind of read a bit about it. I mean, in the OpenWorm kind of mission is trying to do a simulated whole worm, but in DevoWorm, is it a mission on doing that, or you're just quite more of a general analyzing kind of embryogenesis in C. elegans? Okay. Okay. Yeah.
If you get the slides, that'd be great.
Okay. Thank you.
Okay. Bye-bye.
Bye-bye.
Okay, so it sounds like maybe we can start putting things together.
Yeah. Okay. I'll send you the. I couldn't find this computer. I'll send to you afterwards. On the some of the reference that I mentioned.
Okay. Is it okay if we have a copy of your slides?
Yeah, probably. I'll get back to you on that. I'll maybe put it up. I think that Bradley was trying to say that the whole thing will be on video anyway, so you can.
Oh, you recorded it. Now you tell me. Jay, could have brushed my hair.
Yeah.
Okay.
That one kind of quite interesting. And do you think that you guys find the database will be useful for you? Okay. 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 Okay.
Ken.
Yeah.
Ken, I got a question. Are you primarily interested in the database development, or do you have some other personal goals?
I do have some kind of goals in terms of trying to do more understanding and doing more of data-driven sort of biology. So it's how do you have all this data, and how do you, as what you mentioned about deep learning, or not using deep learning but other techniques in order to look at cell biology and as well as in embryogenesis. So my personally, I'm quite interested in how to utilize the database and to look at embryogenesis, or in terms of mathematical biology, or in terms of theoretical biology, some sort of ways in order to develop some sort of how to say, a mathematical model of cell division or embryogenesis. Okay. That's a real quite long-term thing. I mean, like I'm not going to finish or doing anything like that. But I thought it's interesting to have a look into that area. Our lab is yes, it's also similar in terms of looking into using C. elegans as a basis, as a model, to look at understanding different genes and how it affects basic biology, basically. So we are doing actually Kyoda is actually doing what it's trying to do is to to do the genome-wide RNAi knockout, as well as a phenotype. So now we are creating all these images or all this numerical data. So you can actually match the two together. So you can actually do a kind of a feature map of different genes causes what phenotypical problems.
So you could get the whole lineage tree for a given mutant or a given knockout.
Yeah, sort of. Not quite. Yeah. Because I think the data set is still only up to 32 cell divisions. So we're measuring in a very early embryogenesis area, and with that is like what kind of things we can find from the data.
Cells is 32 cell divisions.
Pardon?
How many cells is 32 cell divisions?
It's not. I think no. I don't know.
Doesn't sound right because
sounds right.
no, 2 to the 8th. Okay.
Yeah, I think 2
to the 8th to the 9th, 2 to the 10th is 1,000. So there's only 1,000 cells in a nematode.
No, no, not 32. No, 32 cell division. So only up to 32 cells.
Oh, I see. Not
32 divisions. Yeah, sorry.
32 cells total.
Yeah.
Gotcha.
Yeah. So it's very early embryogenesis.
Now, what's the problem with
compared with Bao? Bao is the whole 900. Yeah, 1,000. Yeah. So 32 cells is very small comparatively. But that's the limit of the DIC.
Oh, it's the limit of DIC.
Yeah.
Oh, I see.
Because you can't see that many afterwards.
How does Bao go too high?
Bao use GFP.
Oh.
So you mark on the every nucleus. But is that, how do you say it? Is that real or whatever? It's a marker.
Okay.
Well, it's a nucleus. It's probably real, but what I'm trying to say is we can't see the other parts, yeah? Yeah. And it doesn't give you that's another problem that we are working some of my colleagues are working on, is to have 2 or 3 markers in order to get the membrane as well as the nucleus, etc. At the moment, you only can, you can't.
I'd like to see microtubules, microfilaments, and intermediate filaments also.
Yeah. Too many things.
And centrioles.
Yeah. Limited to our technology, I'm afraid.
We need a bigger nematode. Well,
yeah.
This is the amphibian cells have an advantage. Because they're very large.
Okay.
At neural plate stages, typically 10, 20 microns by 50 microns.
Okay. Wow.
Single cell.
Okay.
Okay. So they're big. Yep. That's an advantage. Disadvantage is that they're full of yolk, and you really can't see inside.
Oh. So.
That's why we're doing the outside surface. Okay. Okay. So we've tried inside methods. We have done micro MRI.
Okay.
And micro MRI worked to some extent. The magnetic field actually alters development about the 3rd cell division. Okay. We're using 11.7 Tesla.
Okay. Well, but if you mention about calcium, there's another thing about so some of my colleagues also looking at calcium wave on fertilization and development. So just wondering if you use MRI, whether it will actually affect because of the electric field.
It might. It seemed to affect the 3rd cell division.
Okay.
In any case, the major problem with MRI is not that. The major problem was the integration time. It took 20 minutes per time step.
Oh.
So
Yeah.
it was not very fruitful.
Okay.
The other the other possibility is soft X-ray tomography, and that might work. But pretty much for that, you need a synchrotron source.
Okay.
I don't know. You have any spare synchrotrons?
Yeah, we do. We do. Yes, we have. Japan, we do have the synchrotron nearby, not us, not in Kobe, but nearby. We have a synchrotron source somewhere.
Anyone doing
Yes,
embryology?
we can use them. I don't know what is the funding and how to do it. Yes, but as an institute, we do have a synchrotron. But
oh, okay. Well, this is a possibility. With a synchrotron,
get some kind of collaboration funding or something like that.
Yeah, but it is a realistic possibility to use synchrotron radiation to produce soft X-rays.
Okay.
Soft X-rays will give you high contrast with these embryos, especially with the DNA, etc. The nuclei should show up beautifully, and probably other structures.
Okay.
Okay.
But on the other hand, it gives to, I mean, you pretty, how do you say it? What's the advantage of using so much things to look at the, it's
higher resolution. X-rays have higher resolution.
Because of the size of the embryo, you expect that you can see more from this type of embryos, aquatic embryos and things like that, which you can see.
Look, you're limited to light resolution, 0.5 micron, right?
Okay.
And with soft X-rays, one might get very high contrast.
Okay.
Okay. And much higher resolution because the wavelength is shorter.
Okay. And how is anybody doing this in the US?
I think there's a little bit of work. Most of the work I'm familiar with is there were some things that they thought were embryos from China that were fossil embryos.
Oh, okay.
And those were imaged. They were very beautiful, but there's a lot of literature suggesting they weren't embryos at all, that there's some other structure.
Okay.
Problem dealing with fossils, you know.
Yes.
So synchrotron radiation was used for reconstructing them and producing very nice images. Now, of course, there they were fossilized, so you had different contrast mechanisms. But I don't know. I could check what's going on soft X-rays and embryos and see. Okay. Yeah, it's a real possibility, and it may solve a lot of problems if it works, because you could image it live using an intense X-ray source. It's monochromatic.
Okay.
Okay, and with being monochromatic, it means that the absorptions, you don't need any what's called beam hardening corrections. If you have the ordinary X-ray tubes produce a broad spectrum, and it complicates the analysis.
I see.
With synchrotron, you can get very monochromatic radiation, which means you don't have any of those problems. So the software is readily available for reconstruction.
Okay.
Okay, I've worked on that kind of software for decades.
Okay.
So it's not called computed tomography software. Okay.
Okay.
It's not a problem. It's just really doing it and having access to what they call a beam line.
Yep.
Okay.
Yep.
And
I will check, see how whether there is any interest in that area.
Okay. Yeah. You know, I think There's a synchrotron in California where I think some X-ray work has been done on embryos. Well, I'll check the literature.
Okay, yeah, that'd be great. That'd be interesting.
Okay.
Good.
Okay. Well, that was fun.
Well,
You know,
thank you. It's quite interesting. It's the first time I do a presentation online, actually.
Oh, really? Oh, okay. Well,
This is new to me too.
Well,
But,
we ran the what we call the embryo physics course.
Okay.
Starting in 2009, and it ran until basically last year or so. And we ran it in Second Life.
Amazing.
Yeah.
Does it work?
It worked. People get avatars, and they showed slides, and we in Second Life, like you're sitting in a seminar. Some of the avatars were outlandish, but that was okay.
Morph into different shape, I suppose. So yes.
So we had. So I know. So that ran about 6 years, and we might revive it if people are interested. Though I think Second Life it's a difficult medium for people.
Yeah, it's difficult, I think.
Just come in the first time. We had to handhold a lot of people.
Physically or virtually.
Yeah. But it was fun.
Oh,
So
good.
I'll, you know what I'll do? I'll send you the 2L Second Life website.
Yes. Okay. I'll check it out.
Okay. Okay. Good. I guess we're creating enough work for everybody.
Right.
Okay.
Pardon? No problem. No problem. My pleasure. And I'll send you the some of the links that I mentioned.
Okay. Great.
Some things like that. Okay.
Okay. Thanks a lot. Have a good day.
Good night. Bye-bye.
Okay. Well, that was good. Okay.